start. My name is Vitor. I'm a postdoc at College of Agriculture of the Ohio State University. I'm going to present work from my previous postdoc with Dr. Miguel Velasquez and Dr. Jean Chamarin that I did when I was in France, working at INRA and at the University of Montpellier. In this work, we evaluate the use of the approximate Bayesian computation with random forest to infer demography and selection in temporal data. One of the goals of population genetics is to quantify the role of neutral processes such as migration, admixture, drift, and selection in shaping the genetic diversity of populations or population of species. We usually approach this assuming that if there is signal of the neutral process like the mixture, drift, you can see this signal throughout the genome. And if there is a sign of selection, we can find it localized in some regions of the genome. So in this classical view of selection demography, we are assuming that selection, if it exists, is rare and it's strong. So imagine that if we were able to see at the moment the beneficial mutation arises in population, we can see that through time, because of the selection, the allele frequency of this mutation is going to change through time in a kind of deterministic way that's easily that we can easily separate from the background noise of neutral mutation, that they have these random walks that's determined by the randomness of the genetic process, such as Mendelian segregation, mating, so far so on. So in this way of thinking, we can see demography throughout the genome and selection localized in the genome. We devise two main approaches to analyze genetic data. The first one is use a kind of stepwise approach where we first try to identify locals under selection, remove those, and then estimate demography, assuming that everything's there that was left is neutral. And the other is develop methods that are somehow robust or assume that selection is rare and then can be negligible during the process of estimation of demography process. But there's a lot of work coming that came out in the last 10 days, I think, that show that this way of thinking is problematic because selection, if not properly account, bias the inference of demography, and demography, if not completely modeled, can greatly impact the inference of selection, especially increasing the type 1 error for the detection of selection in genome scans. So, so that was the ideal way of seeing selection demography, but the reality is a little bit more complex. The first thing, that selection acting in a beneficial mutation carries neutral mutation together that were really close to that mutation or far from it, because there's a interaction between linkage and the strength of selection that create these blocks that are hitchhiked together during the sweep. So when we try to find those mutations affected by selection, we fail to find all those mutations to remove for the demography inference, assuming neutral markers. And the other thing that I think is more problematic than linkage is the fact that selection of mild and weakly selected mutations are much more frequent. And because the selection is not strong in those mutations, it's really hard to find those to account for in methods or to remove from to estimate to infer demography. So in reality, although many methods perform well when you evaluate under the standard assumption of neutrality, it's difficult in practice to assure that all the nucleotides that we are using, all the data we're using, are experiencing only neutrality because of linkage that selection causes in nearby neutral mutations and because of a lot of weakly to mildly selected mutations are present in our data sets. So today there is because of the advance of sequencing, it's a really nice, it's a really unique time to study evolution because we can use ancestral DNA to learn much more about the evolutionary process. It's a kind of way to look at the past and learn from it how selection and demography shape the diversity of species and populations. So there is a lot of interest in to use this type of temporal data sets to learn much more about selection and demography. One of the early, I think, ways works that showed the use of neutral markers for the inference of population size was came from this paper of Ney Tajima. So they proposed, and at that time we only have few markers, like most of them neutral or quasi-neutral, like allozymes. But we have few markers, and then the idea is to use the standardized variance in allele frequency, like an FST, to estimate the temporal effect population size. And this worked quite well for neutral markers, but when we start having much more markers like SNPs, the chance we are going to sample markers that are neutral or affected by selection is much larger. So in 2004, Goldrigen Battalion suggests to use the F statistic the FC statistics in a kind of stepwise way to first identify locus under selection. It's done by calculating the value for this statistic for each loci and fitting this distribution to a chi-square distribution, and then finding string values. So by removing these string values, in theory, you are removing markers that were get by selection. And then with the markers that were left after the removal, you can estimate the effect population size in a way that it would be only from the neutral markers. But and other, like following this modify use of the FC statistics, there are others that authors that develop a similar statistics, but instead of using the FC, from Tajima, they use the Weir-Cochran FST. That this statistic has much better properties than the FC. But again, doing this kind of stepwise separation of neutral and neutrality and selection has a lot of problems, as I presented before. So that kind of thing that motivated us to try to come up with a way to estimate these parameters accounting for the selection. So we propose to do a joint inference of effect population size and selection using ABC random forest from genome-wide temporal data. So we approach this by first defining a model that's going to be our working model. We start very simple. So we start with it's a model of one isolated population that has any one individuals. Each individual has a genome size of G. And then in this population, it was sampled in 2 time points, T1 and T2. We simulate the dynamics in a Fourier-time way of a drift selection equilibrium. Drift was determined by the random sample of gametes and the Mendelian segregation recombination, and selection was determined by the influx of beneficial mutations. So we just model selection acting on de novo beneficial mutation. So because it's a Fourier-time simulation, we need to have a burning period, to make sure that the initial conditions of the simulation were removed by the time we had the sample of individuals. The way we model selection and was dependent of the way we model the genetic architecture. So we decide to be very simplistic in this way. We decide to take the haploid genome and split this genome in neutral and non-neutral regions that has the same size. For the proof of concept, we decide to have all the fragments on all the regions with 50 kbp. So why we decide to split the genome in 2 parts? Because it was easy to define values for two parameters that we can implement in our prior sampling for the ABC. So we define the first parameter, that's the proportion of regions that are non-neutral. So that's it's a fancy way to say, okay, how many fragments are going to be the one that's going to hold beneficial mutations. And the other parameter is define how many mutations that are non-neutral is going to be present in these fragments that are non-neutral. So we have 2 parameters that combine with the mutation rate of the whole simulation, define the mutation rate of the beneficial mutation. So for each mutation that is beneficial, a value for the selection coefficient is sampled from a gamma distribution with mean gamma and scale parameter. This was defined for every simulation has its own gamma distribution that the value of selection coefficient were sampled. So at the end, we have a very simple model with 6 parameters. So we have like we define a mutation rate that's going to well, we have a mutation rate that's going to be for the whole stimulation. So we don't have different mutation rates in different parts of the genome. We have a one parameter that defines the genome-wide recombination rates. We have the population size for the burning period and population size for the inferential period. The mean for the gamma distribution in this compound parameter that define the number of mutations that are non-neutral in the simulation. I decided to put this in a table to show that in theory, we can use all these model parameters to make inference. If in ABC, assuming that all these model parameters is a dependent variable. I'm gonna show this, it's not a good idea because some parameters cannot reflect the stochasticity of the selection dynamics. So I'm gonna introduce something that was what we use for making inference about demography and selection. That's the latent variable. So latent variable is defined as a variable or quantity that is inferred from other quantities. In this case, latent variables in the ABC framework, we developed these latent variables kind of capture they emerge as values that capture the selection and drift dynamic. And one of these latent variables that we define is the effect population size, defined as the variance of reproductive success of individuals. So in every simulation, in every generation of the inferential period, we calculate the NE for that generation using from the N of the population size, and the variance of the number of gametes from the parents from the previous generation. In a population equilibrium, the variance of the number of gametes is close to 2, which makes NE close to N. But in reality, this variance of number of gametes is never going to be close to 2, which makes NE different from N. And then, so we did this we calculate this NE for every generation. And then for the whole period, we have a NE that's the harmonic average of all values. And that's the one we use to make inference to estimate the effect population size with the random forest. We also define other 4 latent variables that describe selection. The first of these is the scale mutation rate of beneficial mutation. I'm going to explain a little bit later how those parameters, how these latent variables were calculated. But for now, it's enough to show first, and then later I'm going to explain a little bit better. So the other one that's also was very useful and very informative about the dynamic is the proportion of polymorphism under strong selection. The third one is the substitution load, and the fourth one is the mean of the selection coefficient of the of the strongly selected mutations. So at the end, we have these 4 latent variables that characterize selection and the effect population size. So and then we use this model to produce simulations to make inference about these parameters using ABC with random forest. So ABC is an approach that's very known in population genetics. With ABC, you substitute the likelihood of a Bayesian inference for simulations, with simulations. And then you use summary statistics calculated on each simulation to approximate so if you make approximation of the data through the use of summary statistics, in theory, it requires a lot of number of simulations. And for neutral models, it's usually done with fast coalescence simulations. But to model selection, we need forward-in-time simulations that's usually agent-based. So these type of simulations are very slow. So one of the advantages of using random forest in ABC is that you don't need to have a lot of simulations to have the same level of confidence in your estimates, which alleviates a little bit the computational burden of using individual-based simulation. And the other thing that's very interesting about the random forest, we don't need to pre-select an informative summary statistics to make inference about the parameters we want to infer, because it's done by the random forest in the process of growing the trees and training the model. I'm going to explain a little bit about how it works in practice. So for ABC with random forest, we need this so-called reference table. So reference table is a table that has in each row you have simulation, and in each row is a combination of the dependent variable, the variable you want to infer, and a vector of some summary statistics calculated for that simulation. So in the results I'm going to show, I have a reference table about 55,000 simulations that has more than 100 summary statistics, as I remember. And this is for the simulation part, the data you don't have, like the data you need to use for training. For the data you have, you only need to have a vector of summary statistics. So other thing that's very important to say about random forest is the also so-called out-of-bag estimates. It's a kind of fancy way to say it's a bootstrap estimates. Because in random forest, each forest is composed by, let's say, thousands of trees. Each tree is trained by selecting simulations and summary statistics. So if you are going to train the random forest for the effect population size to learn how to predict the effect population size, you're going to have like thousands of trees in a random forest that's going to use, let's say, 3 of these simulations or 4 for the other summary statistics and so far so on. So at the end, you're going to use part of the data to train the model, not all the data. It's important to stress that because with a reference table that's composed around 50,000 simulations, you don't need to split this reference table for training or for testing. You use the whole reference table to train a model, because a lot of these simulations are not going to be used to train a model or to train a random forest. In this way, you can use the same data to have the prediction of the dependent value you are trying to learn. I'm saying that because most of the evaluation of the random forest I'm going to show going to be a scatter plot with where I show the true value of the parameter, the value that was produced by the simulation, and the estimate using the estimate obtained with this out of bag sampling. So the nice thing about the scatter plot, you can see all the data, and it's very useful to look at this distribution to see how good or how bad was the trained model to infer the parameter of interest. So for a good performance, we're looking for something like this on the left. So the true value of the parameter is clustered together with the estimated value in this diagonal of this scatter plot. And when you don't have like good inference or good training, you see the points spread randomly across the plot. So keep in mind these plots. That's going to be very helpful to evaluate the performance of our random forest. So the first question that came is, okay, with all these and with the ABC random forest, were we able to estimate demography that accounts for this properly accounts for selection? And the answer is yes. Those are the NE estimated with the random forest. So we can see clear that the true and estimated value are very close to each other, clustering this diagonal of this plot in the left. In the right, we have one of the parameters informative about selection that we also train with random forest. It's important to stress that each parameter was trained independently with independent random forests. So in the left, we can see the theta b is somehow well estimated. So yes, we can answer this question. So we were able to have a joint inference of demography and selection with ABC random forests. And the other thing, other question that's also important to ask, how well we did compared to other ways to estimate NE with temporal sample. So as I introduced it before, there is a modified version of the temporal NE that's calculated instead of with the FC statistic, it's calculated with the FST. So if you have FST calculated for 2-3 samples in different years or different generations, you can use that estimated FST and account for the time between samples to have an estimate of NE. So what we did was to take all the global FST that was estimated for every simulation and calculate the NE for all the simulations. So this is what we see here. So in the x-axis is the true NE for the simulation produced by the calculation of latent variable. And in the y-axis is the NE from this equation for all the simulations. So we can compare with our estimates and see that our estimate did a better job accounting for whatever selection is happening. We went a little bit further and asked what is causing this difference between these two estimates. And one thing we did, we calculate the mean square error between the true and the estimated value for both estimators, and plot as a function of the theta b that somehow measure the amount of selection. For the NE from the temporal FST, we see that when selection start becoming very frequent, when the log of theta b start becoming very positive, the estimator did a very poor job in accounting for selection. In comparison, our method suffered from this as well, but much less pronounced than the NE for the FST. Other thing that we could see with this approach, because we model the NE, the population size in different ways, we were able to get an estimate of the true demography when we train our random forest with the population size defined for each simulation. And we have the estimate of the effect population size when we train a random forest with the NE that's a latent variable. Now I'm going to explain a little bit what means each of the parameters of selection. So we have 4 parameters. I will start with the scale mutation rate of beneficial mutation. It's calculated as a compound parameter that also we can say that's a latent variable. That's a product of the population size, the mutation rate of the beneficial mutation is scaled by the genome size. This latent variable can be interpreted as a way to learn about the adaptive potential of a population. So when a population has very small theta b value, it means that this population needs to wait a long time until a new beneficial mutation arises in the population, which means that if something happens in the environment, this waiting time is going to be a limiting factor. On the other hand, if theta b is large, it means that the waiting time for a new mutation arrive is very short. So if something happens in the environment, the population can rapidly adapt to it. So with this in mind, we can say that when theta b is small, population is limited by the adaptation of this population is limited by mutation, and when theta b is large, the adaptation is not limited by mutation. The other parameters, it's kind of complementary, I think, because now we are looking for the adaptive diversity that is present in the population. So this is I should say this is not mutation load, because we are not looking for deleterious mutation. We are looking for new beneficial mutation, so we should say it's a substitution load. So it measures somehow how much of polymorphism or diversity is adapted, and it is present in the population. So if L or the genetic load is 0, it means that there is no polymorphism or beneficial mutation in the population, because there is no diversity based on the, let's say, the low adaptive potential of the population, or all were lost at that time, or if the influx of beneficial mutation is large and a lot of those mutations are strong selected, it quickly fix this beneficial mutation in the population. That's the other reason that the load will be close to 0. On the other hand, if L is close to 1, it means that there is a lot of adaptive diversity in the population that can be used for to adapt to new environments. So they kind of are complementary if you know, to characterize the adaptive history of a population. The other parameter that we also were interested to estimate is the proportion of mutations that are present in the population that were strongly selected. So remember that for each simulation we have a gamma distribution of the fitness effect. So we, from these mutations that are in population, so we're in population that has a selection coefficient high, like it's the it's higher than this ratio of the population size. It means that there are very strong selected mutations. So we can calculate those from each. We can calculate those on each simulation and divide by the total number of segregating mutations at that time. So in this way, we are considering not only mutations that are present from the beginning to the end, but all beneficial mutation that enter, were strongly selected, were fixed, or were lost for some other motive. So in a way, it kind of complements the other two statistics by showing how much mutation are under strong selection, or what the fraction of the genome is affected by selection. And the last parameter is the average among all the selection coefficient, but not all for all mutations. It's only for the mutations that are strongly selected. So as I said before, for each simulation, we have a gamma mean that defines a gamma distribution. From this distribution, we sample selection coefficient for each new beneficial mutation. But most of these mutations are going to be lost because they are weak or mildly selected, and they're not that informative about the selection dynamics. But if we look at those mutations that are really strong selected and take an average, we might end up having something like this, a version of the gamma distribution that really accounts for those that are being selected for. So we decide to calculate these and then make inference of this parameter. We ended up having not a good, like, compared to all the other two parameters, the theta b and the proportion of strongly selected mutation, it worked not so good, but still we can see that for when the dynamics is pretty high, we can have a good estimate of this parameter. I also should explain that why we have these bar plots. So for these parameters, the proportion of strong selective mutation, this average of the strongest selective mutation, the genetic load, we need to remove neutral simulations to train the model. So after training the random forest without neutral simulations, we went back and calculated these parameters for those simulations, and we can see that we have a lot of bias because of the neutrality. So, so if your system has it's close to a neutral dynamics, we're going to have a bias because the random forest was trained when selection is frequent. So to summarize this part, I would say that we have four estimate of selection. We don't need not more than the polymorphic data to make inference about these parameters. So this is good, especially for non-model species, because we don't need to rely on annotation or other sort of information to learn about these parameters in these species. Another thing I would stress is the use of latent variable instead of model parameters. So I highlight here three pairs of model parameters and latent variables, and I'm going to show that, especially for selection, latent variables did a better job in characterizing the selection dynamic. The first thing to say is that the mutation rate wasn't our target, but with this approach, we can estimate the mutation rate for the time between the two samples. This is a mutation rate assuming that this is constant throughout the genome and throughout the time. So care should be taken when you use this parameter to learn about this in real data sets. But for the recombination rate, we cannot use, as it's similar to a random distribution of points that I showed in the beginning. For the pairs, I want to show the use of latent variables. The proportion of strongly selected mutation as a latent variable is much more informative than the combined parameter that shows the amount of mutation under selection calculated with the parameter that define the number of regions and the number of mutations in those regions. And similarly, the average of selection coefficients of strongly selected mutations is much more informative about the dynamic than the model parameter that define the gamma, the mean for the gamma distribution. So this is for the proof of the concept. So we could see that with this approach, we were able to estimate the population size and characterized selection in temporal data using ABC random forest. Then we decide to test our approach in a real data set. So we found a really interesting data set that was published by Quidland and collaborators in 2018. They have a lot of samples from feral honeybees and also from domesticated colonies of bees collected in California, and they sequenced the whole genome for all the samples. It's important to say that honeybees in California has a mixture of lineage. The M lineage from the Western Europe was first introduced in the US in the 17th century. And then only in 1853, they were introduced to California from US colonies. The Italian C lineage was subsequently introduced in 1859. And more recently, in 1956, from introduction of Africanized bees in Brazil, those bees ended up arriving in California. So, so the domesticated colonies would be a combination of these three lineages, and also the feral population, the population that were that start from domesticated bees that escape and start colonies, could be a mixture of these three lineages. It's important to say that this data set, as I said, contain a lot of other data that we didn't use. We just focus on the populations or localities that we can have both modern samples or museum samples. So in this data set, we found 7 of these populations that have this pair, and those that we use to test our approach. It's a very interesting data set because we have a lot of different time span between pairs, from 15 years, that one in the Placerita Island at north. And then also we have pairs that span 104 years. So what we did, we took this data, combined the modern and museum samples in the same VCF, calculated summary statistic with EGLI, similar that we did in the pipeline for the proof of concept. Produce a vector of summary statistics for each of these temporal pairs, and we simulate for each population. We will obtain for each population a reference table simulating the genome size and the time span between samples. We use, for the mutation rates, we define an informative prior for this, instead of using a non-informative prior that we did for the proof of concept. For the proof of concept, we use a uniform prior. I forgot to say that before. But for this, we define a informative prior setting a normal distribution with the mean as the mutation rate estimated for Hymenoptera that was published in 2017. For the population size, we define a uniform prior from 1 to 10,000, and produce and run simulations to obtain a reference table for each. So we run about 15,000 simulations for each pair of population. So that's the data we use to train a random forest for each population, for each parameter. And then we use that trained random forest to predict parameter when we have observed data set. In this case, the vector of summary statistics for each temporal sample. So again, for the population size, we did the ABC random forest was able to infer quite well the population size for all the populations. The parameter of selection, it shows that selection is not the proportion of the scale mutation rate of beneficial mutation. So that its value is very small. But with this plot, we cannot see clearly because there's a lot of variation around this estimate. But when we use this plot and use this result and combine with other two estimates of selection, we can see that selection is very rare, as the proportion of strongly selected mutation is showing that selection is very, very rare in the population. There is a kind of almost a negligible amount of selection in the population for all the population. And it also said, you can also see this with the genetic load that's showing that there is almost no adaptive diversity segregating the population. So we can conclude that in this system, adaptation is limited by mutation, and there is a negligible amount of strong-sided mutation in the population, as expressed by the low adaptive diversity of those. So it might be because the changes in beekeeping practice that happened in the last 50 years in California, with the changes in proportion of the bee lineage, or it might be in response to the introduction of Africanized bees that increase the admixture, but it also could cause breeding depression in those populations. And another thing that's very important to say that it could reflect the pressure imposed by the introduction of the verroamide. The verroamide is a pest. It's the most important pest of honeybee colonies. So it could increase drift and impose selection in a way that only those that adapt were compose the current population. But also could be because honeybees, as other social insects, they have something that's kind of a vertebrate-like population size, as reflected by the low genetic diversity, higher ratios of non-synonymous mutation changes, which are comparable with larger vertebrates. So we don't know exactly what was the cause of the low population size, but somehow a combination of those factors. And other thing that's interesting to say that because selection was not so widespread, these in part explain the population size close to the census size. We have similar estimates for both parameters for almost all populations, but also to reflect that for a good inference on the census size, you should simulate the life cycle of the bees closer to what is in nature, or include some features of the life cycle that we didn't include in our simulation. So, and the last thing I want to say that, which complements what I said about the other parameters, is the mutation rate. We saw a larger mutation rate compared to the one that was estimated for the Hymenopterus. It might reflect actually the amount of admixture that happened in those populations during the time period. So we can see the curve of the mutation rate was much more far in the left than close to the normal distribution of this as the value that we use for the prior, that the distribution they use for sampling value for the prior to simulate this mutation rate. So in this plot that was present in the paper, we can see that through time, the proportion of each lineage in each population change, which might reflect be a product of the admixture that change as well. So we have this for, let's say, the in red here, the Avalon population. The museum samples has much more composition of the M lineage than the modern sample that start having more C lineage ancestry. And it also can be seen in the placenta in green at the bottom right of the plot that start having much more larger contribution from the Africanized bees in the modern sample. So to conclude, in summary, with this work we show two technical novelties. One is the use of whole genome individuals in the ABC framework, and the other is the use of latent variables as a way to learn about the selection and demography. I also can say that with this approach, we were able to infer demography correcting by the effect of selection. And we were able to estimate 4 parameters informative about selection without using any other information except the polymorphic data from population, from marker data. We have, I also should say that we have a lot of things to work. There are some limitations that we need to work. The first thing is this is a very simple model. It's clear with the analysis of the B data that we should include admixture in our model to accommodate other evolutionary forces. For the moment, it's only possible to work. We should have 2 time points, but Miguel is working in extending this approach for and generalize it for more than 2 time points. Our model only considers selection acting on de novo mutation, beneficial mutations. So it might be interesting to include not only positive sweep, but background selection. It also be important to include selection acting on standard genetic variation. That seems to be the most frequent type of selection happening in real populations. And another way to realistic, because we need to include all these things in a way, so a better way to more to realistic model the genetic architecture and the distribution of fitness effects. And the last thing I should say that this approach, the way it is, it's very computational intensive because it's for time simulation, and I'm simulating the burn-in with this approach. It takes a lot of time to produce simulations, but again, Miguel is working in a version of this pipeline that is going to be a hybrid approach of coalescent with coalescent and forward time simulation that seems to speed up a lot the production of simulations for the AGC approach. So with this, I'd like to thank Miguel Navasquez and Stefan Ermita, especially Miguel, that was my supervisor at the time I was in Montpellier. And I also like to thank the source of funding for this project and the staff that helped me set up the working space to run simulation in the server. And with this, I like to say that if you have any questions, please you can send me an email or reach me out on Twitter. If you are interested in learning more about this work, you can find it in bioRxiv. And with this, I like to take some questions.
Thanks, Vitor. That was a great talk. Really enjoyed that. And congratulations on this work. And I guess, is it in review, or are you still working on it and developing it further at the moment?
I'm not working as I was working when I was in this postdoc. Miguel took from where I stopped and then started working in a way to speed up the simulation, especially in this part, and also to include more time points. So I don't know if we're going to include this in this version of the manuscript, or it's going to be another work that Miguel is also working on.
Okay, cool. So what we've done for the past videos the seminars that we've had on this ADAPT series is ask 3 questions at the end, and I've kind of scribbled down 4 actually. So I'm going to cheat and do 4 questions, and they're kind of just simple ones really. But I guess what most people are working with real data is probably quite small sample sizes. If it's natural populations, they're going to be going into museums like you did with the honeybees, for example. Potentially working with single figure samples, and it looked like you were getting useful data from like even sample sizes of just two individuals from museum samples from some populations, or did I misread that table?
Yeah, I was quite impressed that we learned a lot with data for the honeybees, because as I can say here, for some of the pairs, we have like 2, maximum 5, 6 museum samples. And then for those populations, it worked quite well. I didn't see any difference in the sample size or for the NE estimate, for example. So, but I don't know much more than this. If, for example, using only one genome, you're going to have a decent estimate. I'm not sure about this, but because it's going to be a different set of summary statistics that we need to include. So I'm not sure. The method we test with samples of hundreds of individuals in each time point, and then we test the model for the bees with 2 to 5 samples. So that's something I've also forgot to mention. When I set the model for this real data set, I need also to work. So we simulate larger populations, and we sample these samples, this number of individuals for each single population. So that's something we need to accommodate in order to calculate summary statistics, to make sure we are accounting for the any bias from this sample of individuals. So we didn't start simulating 2 or 5. We start with a larger population, then we took 2 or 5 individuals.
Yeah. Okay. Cool. Well, it looks really promising, and I guess one of the other things that's an issue with real life populations as well is they're not isolated. They don't really conform to these kind of simple Wright-Fisher type model systems. And that's obviously a problem with a lot of different, or at least that's a, what's the word? That's an assumption with a lot of these different methods and models. But it looks like yours is robust to admixture, and you're kind of teasing that out from the change in the mutation rate through time.
Right, that's true. Yeah, that's something that's true. Like when the effect population size, yeah, that's. But I don't know how far it would be robust, but I think it would be interesting to include the mixture in the model, the modeling process. I think that's something that's very interesting to include as well as a research, I think.
And why do you think there are some model parameters that it's struggling a little bit to
Yeah, that's good. That's a
recombination rate was one that really looked strange.
Yeah, that's a good question. I think the dynamics didn't capture, like the effect of the selection and the Briggs dynamics wasn't captured in the summary statistics, but especially for the the recombination rate, we didn't include much. No, we didn't include many. We didn't include summary statistics that are really informative about the recombination. So we didn't include R2 or CZ statistics that are usually used for the inference of recombination. So that might be the case. Okay. Or like the information of the summary statistic between two time points, maybe it's not enough for doing this inference of recombination rate, I think, differently than mutation rate that clearly we could see that we are able to estimate. So I'm not sure. But for the other parameters that I show comparing the latent variable and the model parameter, latent variables were much more, they capture the dynamics in comparison to the thing that has the potential to be there. That's the case for the model parameters.
I wonder if, I mean, I get this has just come to me now, but I wonder whether, because a lot of, it's geared towards de novo mutations. I mean, I wonder whether these coronavirus data sets could be a really good test set, because there you've got one or two mutations and you do have huge changes and lots of speculation on whether this is essentially selection or just demography. Have you sort of considered that?
Yeah, that's true. That's something that's and then this model actually work well in systems that are not limited by mutation, like virus or insects, not bees, but other insects that has a lot of mutation going on. So it might be, yeah, that might be the case for like using and making modification to learn more about the selection dynamic that's happening in the coronavirus. That's a good point. That's cool.
And then just one last question was, so you mentioned that the ambition is to try and basically make it applicable for scanning genetic variation and background selection. And I guess there you could anticipate that you're going to see much more genome-wide selection, more pervasive, I guess. And so I guess teasing that out from potentially neutral evolution that's probably going to be quite a substantial task. Or have you already got some preliminary results that suggest it's achievable?
No, that's something like my postdoc was very short. I was working like 2-3 months before leave in trying to model selection and standard genetic variations and in background selection, but doing it separately. I couldn't finish. I decided to focus on these and have something that's working, let's say. But yeah, that's going to be a little bit complicated, I think. Especially for making inference about. I don't think for the effect population size, because the way it's modeled in our simulation. So DNE just reflects the variance among the genetic contribution from the parents. So either with positive or negative selection, we're going to have this same sort of dynamics that's genome-wide.
Yeah.
But for the population side, I don't think you're going to have a lot of changes, or challenges, I'd say. But for the parameters that describe the selection dynamics, like the distribution of fitness effect, the proportional structure, maybe that could be the case.
Yeah. Okay. I just, maybe more time points as well would be more helpful, I guess, in that. That's more subtle changes in allele frequencies.
Yeah, that's true. And also probably using other, including other summer statistics. We can include other that are more informative about the like recombination, as I said, others that capture the difference between like hard and soft sweep, like the H1 and H2 and the ratio in between these two statistics, and so far so on. Maybe that might be the case, or like including much more or different number statistics for this.
Yeah. Cool. That's great. Thanks so much, Vitor, for your time and giving us this really cool presentation today. And
thank you for the invitation. I really enjoyed doing this.